glass of water.
Yep, can you hear me? Yeah, thank you so much for giving me the opportunity to introduce my project. I'm actually from Finland, and it has been really great to see the PCD diagnostics in the UK and the great family support group, which we don't have in Finland yet. So it would be nice to follow up on your footsteps also in Finland and get the PCD diagnostics going and all the support for the patients. But today I'm just going to introduce to you quickly the project that I'm working on, and my background is in male infertility, and I'm really excited to study the association, the interaction between PCD mutations with male infertility. And why would I want to study infertility in PCD? Of course, it's known that the sperm fertility problems do often occur in PCD males, but the fertility assessment becomes only relevant in the adulthood. But not all PCD patients are affected by male infertility. And actually the female infertility is part of PCD as well, but I'm not so familiar with that, and it is actually even more rare than the male infertility. And the reason why male infertility doesn't occur in all patients is the fact that there are over 40 genes, as Hannah told you, and not all of these genes are important for sperm formation. But so far we don't know which genes are important and which are not. So there haven't been many reports about male infertility or studies how these different genes do affect sperm production. And the main reason why sperm motility may be affected in PCD is the core axonal structure, as Hannah already showed. It's exactly the same along the sperm tail, although the sperm tail also has different accessory structures. So there are differences, although the basic structure looks very similar. And also the movement of cilia and sperm tail, it is different. So we can expect that there are some slight changes how the motility is produced in the sperm tail and in motile cilia. I won't go deep into any genes or protein functions in cilia or sperm, but there are some indications that different proteins are required for the dynein arms, which Hannah already introduced to you. So in sperm dynein arms there are different proteins. If we look at the next slide, in cilia, outer dynein arms, we have DNase 11, DNase 9 and DNase 5, which are very common genes in PCD, but it seems that in sperm these proteins are not as crucial. So they may be not highly important for sperm motility. And when we have been looking at the presence or expression of these proteins in the lung and testes, it seems that like DNase 5 is really not present in the testes, and DNase 8 seems to be testis specific. So, and also the fertility rates for these dynein genes seem to differ, so that DNase 11, there has been reports that the ratio between fertility is 1:1, and for DNase 5 even lower, like 1:4, and for DNase 9 we don't know yet. So there aren't that many reports about the infertility rates. There was a publication in 2017 for 49 men where 25% of the men were fertile, 75 infertile, but there was no motility assessment or ultrastructural studies done in this paper. What we do know about the effects on male fertility, so far there have been reports for 19 PCD genes causing infertility, but there are also some exceptions. like I said about the DNase 5, DNase 11, there seems to be subfertility. For the multicilia genes, MCI does CCNO, they don't affect male fertility at all, and so far all the PCD males with RSPH4A mutations are fertile. And this all is probably because of the different motility pattern of these different cell types. And how does PCD gene mutations affect the sperm? There are different ways because we have so many different genes, and they have different roles in cilia and also in sperm tail. So you just may have lower sperm count or lower motility, so maybe it's just a mild effect on fertility. Or there can be also complete absence of sperm or malformed sperm, and sperm malformation can affect the sperm tail different ways. So let's just really causing short sperm tails or coiled or twisted tails compared to the control. And my research project, it is EU funded and it's running until the end of August 2020. And the aim of the project is to identify the correlations between PCD specific gene mutations with male infertility. And we are recruiting adult male PCD patients to collect sperm samples for our studies. And if you want to participate, what do we expect from you? On the day of recruitment, you will answer complete health and fertility questionnaire, a simple fertility test, just a measure of testicles will be done, and then you would give a semen sample, which is used for quality analysis, and we also store part of that sample for our studies in the lab. And our studies include experiments to identify the cause for the possible observed infertility, morphology, and the studies of protein content in sperm. And what do we exactly do? We collect the information about sperm quality, count and motility to assess your fertility status. Then we use the sample to study the protein localization or presence along the sperm tail with immunofluorescence staining, and then we look at the ultrastructure using electron microscopy. We compare the structures to the control sperm sample and try to see if we can identify any specific structural changes. And in some cases, these changes can be there are maybe more clear changes in sperm tail compared to cilia, so this may give some idea what's going on in motile cilia as well. So we can look at the different, like PCD patients, the lack of dynane arms, or in some cases the lack of central pair of microtubules. And why should you participate? So if you give us a sperm sample and the quality is assessed, then you will get information about your fertility status. This, of course, would help fertility counseling, and also the clinicians can evaluate the required treatments if you want to have children in any point of your life. And of course, then you would help us to understand the fertility in PCD and differences between motile cilia and sperm. And just quickly, there are different treatments depending on the sperm quality. So if the quality is only decreased slightly, then maybe some changes in lifestyle may help you to increase the sperm quality. Or in case the sperm is completely immotile, then the intracytoplasmic sperm injection is possible to help conceive. And in case there is no sperm in ejaculate, then we can take a testicular biopsy and use that for future treatments. And I really hope that we can recruit some some patients to participate in our project. If you are interested, just come and talk to me. You can contact your PCD clinic doctor to get a referral letter for a fertility clinic. So far, the main fertility testing is done at Hammersmith Hospital in London, but we have agreed local fertility clinics can collect some sperm samples, at least in Leeds and hopefully in Southampton in the future. So that would help that not everyone has to travel to London. So thank you very much, and please come and talk to me.